Right, can you see the slide?
Yes.
Physiological basis of herbicide selectivity. Right, what I'm going to talk about is what can happen inside the plant cells. This is just a plant cell so that you know that we're talking about something which is living. So most of the things will take place inside the cell. Whether the herbicide is going to cross the cell walls, once inside the cell walls, what is going to happen to them? In most cases, the herbicides end up in the vacuole after decomposition. So the first thing we are going to talk about is the movement of the herbicides once they get into contact with the plant. So the first thing is that the droplet is going to fall on the plant. So herbicides penetrate faster in young weed seedlings than mature weeds. So depending on the age of the plant, one of the plants may die if it is a young uh, weed seedling. Remember, this is why we say when you are putting post-emergence herbicides, you should do it timely. Apply the herbicide when the weeds are still at the young stage. If you miss it, then you are not going to kill the weeds. And it happens several times on farms where farmers fail to kill these weeds simply because they are just mature during a time of application. The cuticle thickness can increase with age. So the first barrier is the cuticle to herbicide penetration. This can actually cause selectivity. Now we're going to talk of the movement or the penetration of these herbicides. They can be affected by formulations. For example, 2,4-D can penetrate the cuticle readily if it is in the form of an ester. In fact, if you look at 2,4-D herbicide, most of the formulations are esters to enable the herbicide to penetrate the weeds. If it is in an amine form, it may not easily penetrate the cuticle. So 2,4-D, preferably let us use ester formulations compared to amine and salt formulations. So this could partly account for selectivity in foliar applied herbicides. It's because it is based on the cuticle penetration. Now, with the soybean, there's the herbicide known as oxyfluorophane, GOR. Now, it is slow when absorbed in soybean, so soybean may escape oxyfluorophane. I know in Zimbabwe we use this herbicide to control weeds in onion horticultural crops. It's not actually recommended for soybean, although in some countries they use it to control weeds in soybean. Now, in sugarcane, if we use 2,4-D, sugarcane can escape 2,4-D because it is tolerant. But beans can be killed. Now, the reason why sugar can tolerate 2,4-D is because of the slow translocation. But in beans, it is fast translocation. So just the movement of the herbicide is accounting for selectivity. Now, 2,4-D free acid, if 2,4-D is in the form of an acid, it can readily be translocated in the phloem to the meristematic regions, which is the site of action that's causing damage. Now, sometimes we formulate 2,4-D as an ester form so that it gets deesterified to form acid forms. Amine forms can also be deaminated to form acid forms. So, tolerant plant species depend on the deesterification process. If it is slow, then plants are not going to be killed. So the process of converting these herbicide forms to acid forms can bring about selectivity. So this could be the ester form. During the esterification, which is necessary in the plant, it forms the acid form, which becomes toxic, and the alcohol form. So this is actually the esterification of 2,4-D. This is the structure. What is affected is this in red, which becomes the acid. And once you have deesterified it, it then moves easily. Now, during the movement of the herbicides, there's also what we call compartmentalization. Herbicides are restricted in areas of plant where no damage occurs. So the herbicide is stopped somewhere when it is moving. Once it is stopped, it won't reach the site of action where it can cause damage. The examples we have got linuron, which is used to kill weeds. In carrots, in Zimbabwe, we can use linuron. Now, in carrots, the herbicide is activated at the point of entry in carrots. That's how carrots can escape this herbicide. Why 2,4-D doesn't kill grasses? It's because 2,4-D is very slow in movement in grasses, for example, wheat and maize. Therefore, we can use 2,4-D to kill other weeds in wheat and maize, because in the crops, its movement is very slow. This could again be restricted movement due to compartmentalization, or the herbicide is restricted in certain areas. Another restriction, which looks like compartmentalization, is known as sequestration, but this applies to paraquat. In some cases, we know paraquat can kill everything green, but it's not true. Some plants may actually resist paraquat because the herbicide is kept remote from the site of action. The site of action could be chloroplasts. This is experienced in conizer species where if you apply paraquat, you are not going to kill this weed. But sometimes this has something to do with resistance of these herbicides. Too much, high frequency of application of paraquat may select conizer species, which end up not being killed. So this is sequestration and compartmentalization. They are more or less the same. So selectivity could be because of differential penetration, how the herbicide enters the plant, how it moves within the plant, which is differential translocation. It can also be restricted in the plant, which is compartmentalization, or it could be also known as a sequestration. All these processes will lead to selectivity. Now, the other issue now is the altered binding site of action. We are now going to talk about the union between some enzymes in the plants with the herbicide. Because when we apply the herbicide, if it is in the plant system, it's going to meet some enzymes, because this is a living system. So let's suppose we use this model to illustrate what we are going to explain. Here we've got a plant, and we cut a small piece, we examine the cell. We are going to find the chloroplasts, which are very small. And we can use the atrazine as our example. We can represent atrazine with this shape, which can fit into this part, which is the part of the enzyme known as the D1 protein of PS2, or simply the plastoquinone binding site, which is actually part of the enzyme. So in a tolerant crop like maize, atrazine is not going to fit to the plastoquinone binding site of the maize. And the maize will survive, it's the tolerant crop. But it could be a resistant weed. And there's a situation where atrazine will fit into the plastoquinone binding site. Then this is the susceptible plant, which is ideally the wheat. So what we are saying is that the ability of an enzyme to bind to the herbicide molecule will produce death in a susceptible plant. It will cause the plant to die. But if the enzyme fails to bind with the molecule of the herbicide, then the plant will escape the killing effect of the herbicide. So in the following examples, this is the concept which I am going to explore. Binding results in killing of the plant. Lack of binding, then there's tolerance or selectivity. So the herbicide groups which are vulnerable to the binding action of enzymes are the triazines, glyphosate, sulfonylureas plus imidazolinones, allyloxyphenoxycarboxylic acids plus cyclohexene-diones. These are the herbicide groups where we can actually attribute selectivity due to binding of these enzymes within the plants. So these herbicides bind to a specific site of action or a sequence of amino acids on the target enzyme molecule. Now let us start with this group of herbicides, the allyloxyphenoxycarboxylic acids. Now these are vulnerable to the enzyme acetyl coenzyme A carboxylase, which is sometimes known as ACC or ACC case in short, acetyl coenzyme A carboxylase. Now the differences in the structure of acetyl coenzyme A carboxylase in broadleaves and grasses can bring about selectivity. Now in our grasses, there's going to be a successful union of the enzyme and the herbicide. And once that union is there, there's going to be a disruption of lipid synthesis. And the grasses will die because of that union. In broadleaf crops, such as groundnuts, soybeans, the acetyl coenzyme A carboxylase of the broadleaves have been slightly altered in such a way that they will fail to bind to acetyl coenzyme A carboxylase, and they will not disrupt the lipid synthesis. As a result, they will survive the effect of the herbicides. We do have examples of herbicides like Fusilade, which we can apply to soybean or even groundnuts, beans. This can actually specifically kill grasses, leaving the broadleaf crops. We have also Pilot Super. Fusilade is sometimes known as fluazifop butyl. Pilot Super, quizalofop ethyl, holon, diclofop methyl. But I'm sure you are familiar with the Fusilade because it's commonly used. And it is one of the examples of the allyloxyphenoxycarboxylic acids, and the enzyme affected is acetyl coenzyme A carboxylase. Another group of grass killers, cyclohexene-dione herbicides, NAB, cethoxidib, select, clothedium. Probably you are familiar with the NAB, which can be used to kill grasses in soybean and groundnuts. Again, the differences of the acetyl coenzyme A carboxylase in grasses which will bind to nub and select, killing the grasses. Whereas in broadleaves, there's failure to bind to this enzyme. And as a result, broadleaves are not killed. This will cause selectivity. Initially, I used atrazine for the triazines, where we have altered quinone binding site, which can result in selectivity. But mostly, this is accounting for resistance in weed biotypes. It's a form of selectivity, but which is not wanted by the farmer because it brings about resistance, where you have triazines not being, where you have triazines failing to kill the weeds. And if we can move on to sulfonylurea and the midazolinone herbicides, again, there's a special enzyme known as acetolactate synthase, ALS. A change in amino acid sequence on the ALS enzyme can prevent binding of sulfonylurea and midazolinone herbicides. The other name for this enzyme is acetohydroxy acid synthase, AHAS. So let me repeat this. The enzyme can bind to the herbicide and cause death to that plant, which allows it to this process to take place. In the crop, the herbicide will not bind to the herbicide, and then there is the selectivity. Here we've got examples of sulfonylurea herbicides. We have got chlorsulfuron, primisulfuron. These are used in very small quantities. Probably you may know nicosulfuron because it kills chamfer grass. In maize, it is known as accent. What happens is that nicosulfuron is going to bind to the enzyme in the chamfer grass and cause it to die. But it will fail to bind to the enzyme in maize, and the maize is going to survive. And in this case, the enzyme is acetolactate synthase or acetohydroxy acid synthase. Now, in the imidazolinone, where we have imazapa, I know imazapa can kill almost everything. Although we can use it to kill striga in imazapa-resistant maize. Imazamox, which is lynx, which is used in groundnuts. Groundnuts can survive because they possess enzymes which do not bind to imazamox. Imazathapa or pesut, which can be used in soybeans. It means the soybeans do not possess the enzyme which bind to pesut. So these are the imidazolinone herbicides. So we are talking about herbicides which can be influenced by the enzymes which are found within the plants, within the crops and weeds, which can account for selectivity. Now, as far as glyphosate is concerned, scientists using GMOs have actually manipulated some enzymes in plants. We know that glyphosate can bind or can inhibit the enzyme known as 5-enolpyruvenoshikimate 3-phosphate synthase, which is EPSPS. Now, what has happened in GMO is that the scientists have actually altered EPSPS using GMOs in maize and soybean, so that EPSPS, which is the artificial one, is not going to bind to glyphosate. Therefore, glyphosate will fail to kill maize and the soybean. So this is an artificial situation where we've actually interfered with the enzyme so that we bring about selectivity of glyphosate. So this is how enzymes can account for selectivity. Now we move on to herbicide metabolism, which will account for most of the selectivity. Now, if the herbicide enters a plant system, which is a living system, it is going to be degraded either very fast or very slow. If it is fast, it can bring about tolerance in plants. If the degradation is slow, then we'll have susceptible plants. So what we can say is that in crops, the degradation is very fast. In weeds, the degradation is very slow. Now, herbicide degradation, or how the herbicide is destroyed within the plant, will influence its concentration at the site of action. The site of action could actually be the enzyme. So here is the summary of the processes of metabolism which can bring about selectivity. We have got what is called oxidation. Now, oxidation, I will spend a bit of time on oxidation because there are actually about 5 processes under oxidation influenced by an enzyme which is very important, known as the cytochrome P450 monoxygenase. Again, the cytochrome P450 monoxygenase and other enzymes as well will influence hydrolysis. The same enzymes again, cytochrome P450 monoxygenase, can influence reduction. Reduction is just one reaction. Hydrolysis, probably there are 3 reactions, and the oxidation, 5 or 6 reactions. And of course, when these have happened, I mean, these three processes occur in what we call phase one. And there's what we call conjugation, which is quite a number of processes which will form what is known as phase two and phase three. And it is these reactions in total which can explain why some plants can die after herbicide application or survive. So we'll go through. Sorry. Hello. Yeah, good. Ndichazofona ndine lecture. All right. So the herbicides are broken in the plants via three phases. The phase one reactions consists of oxidation, reduction, and hydrolysis. And these are actually catalyzed by cytochrome P450 monoxygenase. Then we've got phase two reactions, which are conjugation reactions. There are quite a number of some enzymes and some molecules which can be important under phase two. Then phase three has got some few reactions which are conjugation reactions, which will move everything into the vacuoles or part of the cell. Actually, this is how plants can protect themselves from pesticides, in particular herbicides. Now, let us focus on the phase one reactions. This is where cytochrome P450 monooxygenase, this is the chief enzyme responsible for reactions which break down herbicides. It occurs in the endoplasmic reticulum, which is part of an organelle of the cell. Sometimes we call them mixed function oxidases. Now they catalyze oxidation reactions. Oxygen is incorporated into the herbicide molecule. This is the beginning of the process to destroy herbicides which enter the plant. So oxygen is used to sort of burn the herbicides. The oxygen atom is converted into water and electron donor. So this is the example of cytochrome P450 catalyzed reaction. Actually, the role of cytochrome is to speed up these reactions without itself participating in this reaction. So we've got number one, NADPH. It's required. Hydrogen ions are required. Then the critical element is oxygen. Then we have the herbicide represented by RH. This reaction will drive to the right. and cause the production of NADP plus water and an altered herbicide, which is now carrying an OH. So RH represents the herbicide, ROH represents the herbicide metabolite or change the herbicide. Now, this reaction is not limited to herbicides alone, but any other pesticides or any foreign substances which enter the plant, which we call xenobiotics. So it's a process which enables the plant to get rid of toxic substances. And of course, it will bring about selectivity. What I'm going to do now is to show you some important parts of herbicides which will be altered by cytochrome P450 catalyzed reactions. Remember, cytochrome P450 is aiding the addition of oxygen to the herbicides so that we get an altered herbicide. Now, we know that herbicide molecules are so complex to the extent that we are not going to remember the structures. But what we want to find out from those structures are these: OH, NH2, SH, COOH, CN, OCH3, CH3, SCH3. These are some of the critical groups which we call functional groups. So if we are able to recognize or to identify functional groups on a complex molecular structure of the herbicide, we may be able to tell how that herbicide is going to break down. Because these are the important functional groups which will facilitate the herbicide breakdown and reduce phytotoxicity. So what I'm going to do, and I'm going to put a red flag on the oxidation reactions. So I'm going to talk about the oxidation reactions, and please pay attention now. The oxidation reactions are simply adding oxygen to herbicide molecules which have entered the plant. And we've got 5 oxidation reactions. There could be 6, but these are 5 important ones. Number 1, we've got what is known as hydroxylation. Number 2, we've got N-dealkylation. Number 3, we've got O-dealkylation. 4, oxidative deamination. 5, sulfoxidation or thioether oxidation. And please note, all 5 processes are adding oxygen to the herbicide which has entered the plant system. Depending on the herbicide, one of these processes is going to be dominant in destroying the herbicide. So let us look at the first one, hydroxylation. Now, in hydroxylation, the herbicide should carry a halogen, a chlorine molecule. This is an aromatic group or a benzene ring, but what will cause hydroxylation is the presence of chlorine. It could be what is known as the aliphatic group, where we represent it by RCl. It's the Cl which will trigger hydroxylation in the presence of this enzyme cytochrome P450. And the hydroxylation is basically this reaction. And we have simplified it. What we get is the OH attached to benzene ring, or the OH attached to R. Once we get this situation here, the herbicide ceases to be toxic. And we can apply this to basagran. This is the structure of basagran. At one point of the benzene ring, we can insert the OH and cause basagran to be less toxic. Or we can just represent it by this circle and write basagran, hydroxylation, we put the OH, and the basagran herbicide properties will be removed. That is hydroxylation. N-dealkylation requires the presence of this structure, RNHCH3. After N-dealkylation, we get RNH2 here. And the herbicide ceases to be toxic because of this change, which is catalyzed by cytochrome P450. And this can happen to atrazine, where we have this complicated structure of atrazine, and this part can be converted to NH2. N-dealkylation, it means altering this or removing this group at the position N. Or we can simplify it like this, where we convert the whole thing to RNH2. This will be atrazine. Then when it is in this state, it's no longer toxic. Then we have O-dealkylation. O-dealkylation should have oxygen here and this methyl group. Now we will remove this CH3 during the O-dealkylation process, and we end up with the ROH, which is less toxic. And we can apply this to the real molecule, which is 2,4-D acid. And it can happen here where you have the oxygen, and you end up with an OH, which is less toxic. Then we have the process known as sulfoxidation or thioether oxidation. For this to take place, there should be an S, a sulfur, attached to R and CH3. Then if we add an oxygen to S, it becomes sulfoxidation or thioether oxidation. And it is facilitated again by this cytochrome P450 monoxygenase. And it can happen to this prometrine, which has got an S. In fact, any molecule carrying an S, we can predict that sulfoxidation or thioether oxidation is going to take place. This is simply addition of oxygen at the position S, like what has happened here. We also have sulfoxidation or thioether oxidation on EPTC. It looks complicated, but let us focus on here where we find S, because that is our prime interest. Then if we add O to S, we have achieved the sulfoxidation, and the herbicide ceases to be the herbicide. It's no longer toxic. Oxidative deamination, it's another process. Now, in order to recognize where this is going to take place, you need to know where there's RNH, then NH2. There have to be two N's which are joined together, double N on the molecule. Then you have an amine being formed. This is oxidative deamination. This can take place on SENGO, which is metribuzin, which can be converted to this NH2 here on the ring. If you look for interest sake, you can see there's an S there, CH3 there, which means metribuzin can also undergo sulfoxidation or thioether oxidation where oxygen is added here. Right. We move on to reduction. Reduction is just one reaction. And for it to take place, we should have an NO2. Then this is converted to RNH2. If we cite a molecule known as trifluorine, which is this one, which has got this NO2, it can be converted to NH2. And this is a nitro reduction. Or we can simply represent this by this symbol R joined to NO2 being converted to NH2. And of course, if you look at this molecule, you'll find that it's also vulnerable to N dealkylation at 2 points, which we have discussed. Hydrolysis, this is the reaction of the herbicide with water. Herbicide with an ester, nitrile, or amide groups can undergo hydrolysis. Right, we have got ester hydrolysis. Here where we have CRC, we put oxygen here, oxygen there, and R there. We can split it to an acid and an alcohol, which is ROH. Well, this is the 2,4-D methyl ester, which can be split to form an acid and the alcohol. I've talked about the de-esterification when I was talking about the movement of 2,4-D methyl ester within the plant. We said that if you have a methyl ester, it can cross the cuticle very fast. Once it is inside the plant, it has to be de-esterified to produce an acid so that you can kill the weed. Again, the speed of de-esterification to produce the acid may cause selectivity. Amide hydrolysis. In amide has got RNH, then you have got carbon attached to oxygen, and then to R. This will form an NH2 here. And we can apply it to a real herbicide propanil, which has got this amide, which can be broken down to NH2. What is important is to be able to recognize these key structures, and you can tell what can happen to the herbicide. The other structure which is conducive to hydrolysis is what we call the cyano group, which has got a triple bond, carbon joined to nitrogen. Now, the cyano bond can be broken to an amide, then to an acid and ammonia. Now, this can happen to cyanazine or Bladex. Bladex can be used to control weeds in maize. And if we look at the structure, we just go for the CN, a C attached to N, where there's a triple bond here, the cyano group, which is converted to the amide. Then it's later converted to the acid. It can also happen to bacterial. This is the herbicide for wheat. We'll just go for the CN, where we have this triple bond, which is converted again to an acid. So this will be then phase 1. Now in phase 2, we have what are known as conjugation reactions. Let me just highlight at this point that I am going to talk about the activity of glutathione. Then I will talk about the activity of sugar, which is produced in the plants. Then I will talk also of the activities of the amino acids, special amino acids produced in the plant. So let us focus on glutathione under conjugation reactions. Now, glutathione is very important for cleaning pesticides which enter the plant system, including herbicides. So it is the glutathione system which is effective in protecting the plants. Now, glutathione will be attached to the herbicide metabolites derived from phase 1 reactions. You know, all the activities of the phase 1, those products will be further acted upon by glutathione. Or sometimes it can go directly to the herbicides without alteration of phase 1, depending on the structure. Now, the enzyme now here in phase 2, which works in conjunction with the glutathione activity, is known as glutathione S transferase, GST. What is reactive on glutathione is known as the SH component. So glutathione is a cleaning substance. It cleans up these herbicides which enter the plant system. It is a tripeptide of 3 amino acids. So what constitutes glutathione are the 3 amino acids: glutamate, cysteine, and glycine, which we can write in short as GLU attached to cis attached to gly. Now, it is the cysteine which holds the SH reactive component. So I can write it in short here to represent a glutathione. GLU joined to cysteine, joined to glutamate, joined to cysteine, joined to glycine. These are amino acids. But at the center here, cysteine holds the reactive component SH. So this product of phase 1, the herbicide which has been given an OH, will further react with the glutathione. This bit is going to get attached to the SH like here, right? So the herbicide is now joined via S to cysteine, which is part of glutathione, and you have water here. Now this bit will cause the herbicide to be less toxic. So you've got glutathione conjugates could be transferred to the vacuole. So this part here will be moved to the vacuole of the cell. And that's the end of the herbicide. Well, if we had the atrazine with the chlorine here, glutathione could go for this chlorine and form this conjugate, which will move to the vacuole. So this is how glutathione cleans the plant of atrazine. In fact, maize uses glutathione to detoxify atrazine very fast. That's why maize is not going to be affected by glutathione. But sometimes we have maize inbred lines with less glutathione, which can get affected by atrazine. Sugar, we know that sugar provides energy during respiration, and it's a product of photosynthesis in plants. It can also be used to clean up pesticides or herbicides which have entered the plants. So we have herbicide metabolites with the following functional groups. If the herbicide carries an NH2, N-arylamine or an amine, carboxylic acid, COOH, or phenol OH, it's vulnerable to sugar activity. So the above metabolites can be converted or can be made into sugar conjugates, and they will become less toxic because sugar will be combined with these herbicide molecules or metabolites to form less toxic substances. Now, where do we get our sugar from? It comes from what is known as uridine diphosphoglucose or UDPG. It gives up the sugar and it's the sugar donor. And we also have an enzyme which facilitates reactions, which is known as UDP glucosyltransferase. Now, when we were talking about glutathione, there was a glutathione transferase enzyme. So UDP glucosyltransferase is the enzyme which is needed for these reactions. We also need UDP glucose as the sugar donor. This then is an example where we have a herbicide altered in phase 1 with an OH. Then we have got UDP glucose donating glucose to this reaction. And we end up with UDP, which is free of glucose. Now the glucose is now joined via the oxygen to the herbicide part. And we call this O-glycosylation. That is the process. And this is the glucosyl transferase enzyme, which will facilitate this reaction. This can happen to Basagran if it carries an OH. It can combine with glucose to form this structure, which is moved to the vacuoles. It's the UDPG, which can actually donate the glucose, and you have UDP glucosyl transferase, or just glucosyl transferase enzyme facilitating the reaction. We can also have glucose acting upon the herbicide with an amine with this group to form a conjugation. So we have UDP glucose. Glucose is donated. It combines with this part of herbicide. Now we have glucose N herbicide. And this is now free UDP, or uridine diphosphate. Then you have got glycosyl transferase, which is the enzyme facilitating this reaction. And the third reaction is where we have the ester. We have the herbicide carrying the COOH. Again, glucose is added to that group to form glucose, and we have C attached to oxygen, bond forming this herbicide. Then we have got glycosyl transferase in all cases facilitating the reaction. This is going to be moved to the vacuole, and the plant will become safe. It's not going to be killed by the herbicide. If we had 2,4-D acid, it can be detoxified via the glucose donation from this UDPG. So we have this very complicated structure which is moved to the vacuole. Then the amino acid conjugations, we've got the herbicide with this acid can react with the aspartate and form herbicide aspartate. It will have this bond where you have carbon joined to oxygen, then NH. We can have 2,4-D, which is an acid uh, being combined with the aspartate and form this complicated structure. So amino acids, especially aspartate, can clean up herbicides carrying COOH. Then we have got phase 3 reactions. Now, plants do not excrete herbicide metabolites. They go to the vacuoles. So further conjugation of phase 2 products can occur. Then moving metabolites into the vacuole for storage is the next step. Or these products can be incorporated into the cell region. They can become part of the cell walls. Herbicide glucosides, addition of more sugar, can be further processed via addition of malonic acid residue to sugar conjugates. Right? Here, it's only one reaction in phase 3. So there are not many things. What you now need is an altered herbicide carrying glucose, like glucose or herbicide. You will have malonate and the malonyl coenzyme A. So you will have the malonyl component added to glucose. Then you have malonyl glucose herbicide. This can become part of the cell walls, or it can go to the vacuoles, this product. Then coenzyme A is released. So that will be the end of the herbicide. Or you can have more sugar being added to form glucose conjugations. So you can have glucose plus glucose herbicide. You get glucose joined to herbicide. This product can be moved to the vacuoles, or it can just become a part of the cell wall. Now I will summarize everything. Physiological basis of selectivity. Now the key points is that you need to, we need to understand the cuticle effects, because if the cuticle cannot allow the herbicide to go in, selectivity can take place. Then herbicide movement in the plant, compartmentalization, sequestration. Now these 4 key points can explain also selectivity. Then the enzyme site of action, or altered site of action, or changes on the structure of the enzyme could also explain selectivity. Here we have got triazines, where we have plastoquinone binding site. This is part of the enzyme. Glyphosate EPSP synthase, which we have actually manipulated so that we can achieve selectivity in GMO maize and GMO soybean. Allyloxyphenoxy carboxylic acids and the cyclohexanediones. We have acetyl coenzyme A carboxylase, ACC. Sulfonylurea and imidazolinones, acetolactate synthase, ALS. Here, mostly we are concerned with the grass killers, grass killing herbicides. This is where the altered enzyme come into effect things like fusilade, nab, just to mention the two. Then when we went on to talk about metabolism itself, we said it can bring about selectivity, where we have three phases. Phase one, we have got the activity of cytochrome P450, and for it to uh, assist or speed up reactions, the herbicides should carry the functional groups, for example, these ones. Then the processes which we have discussed: oxidation, we have got hydroxylation, N-dealucylation, O-dealucylation, oxidative deamination, and the sulfoxidation. Then we have got a reduction. Sulfoxidation sometimes is called thioether oxidation. Then reduction, we have got NO2. Hydrolysis, we should have esters, amides, and nitriles. Then conjugation reactions. Here I am going to lift a red flag, right? Where we have glutathione, which is glutamate joined to cysteine joined to glycerate, which has an SH component. And we've got glutathione S transferase, which will facilitate the reactions. So here I have raised a red flag. UDP glucosyltransferase is the enzyme which is important. UDPG can produce glucose, and it is glucose which will join the herbicides with OH, NH2, and COOH. We also have aspartate, which is an amino acid, which will react with such herbicides. Then, of course, we have malonate, which will produce malonyl, which will join to glucose herbicide from phase 2. We have malonyl coenzyme A, which produce malonyl glucose herbicide. Then the last, we can add glucose again in phase 3 to form glucose added to glucose herbicide structure. This is what we call a GND biocide. Once we have done that, the herbicide will have been destroyed. So in order to produce selectivity, it's the rate at which these reactions are taking place to destroy the herbicides, which is quite a long story. I'll end here